on the last session, I thought we would just change a few slides and get the hepatologists to refocus, to not bring us out of the dark ages, so to speak, but to sort of come forward from 65 million years ago to today. So I will get to my topic in a minute, so don't worry, Harry. But I do want to sort of make a few points in relationship to what was said in the closing sessions of the previous session. So what's in a name? Well, this is what was in discussion really was the natural history 4 phases of chronic hepatitis B, which is shown for you here on the immune tolerant, immunoelimination, immune tolerant, immunoelimination, and then latent. So if you're an immunologist, I mean, what is immunotolerant anyway? You're tolerant to the organism. This is actually based on not even a quantitative E antigen level, but an E antigen positivity level, near normal or normal ALT, with an ALT varying between 2 to 3 times the upper limit of normal, depending on the laboratory you use anyway, and high DNA. High DNA, most people don't even titrate out their DNA, and if they use the Roche, earlier Roche assays, they just got greater than 10 to the 8. So why that's important is because it affects the clinical practice to guidelines. So if you're in the immune tolerant phase, as we discussed, you're excluded from treatment. If you're in the immune elimination phase, and so what's in the immune elimination phase is something magical happens, something wakes up. There's a switch that goes from the immune tolerant to the immune elimination. But I mean, both words are really meaningless to a virologist because the host doesn't actually get rid of the virus. It's not immunologically eliminated. It's still there. And so what the immunologist said, we'll call it latent. So latent is a phrase that we don't use in hepatitis B really, because if you have a decent enough DNA assay, most patients are DNA or viremic anyway. So the word latent is really quite inappropriate as well. Now that's if you're in the E antigen positive phase transitioning. What happens to that subgroup of patients that transition to E negative? 20 to 30% of our chronic carriers. So we've got immunotolerance and immune elimination, a brisk or brief latent phase, and then the word reactivation appears. Reactivation implies that you go from a state of negativity to a state of positivity. Well, that doesn't really happen either, but when you morph from one to the other. And again, my point being is that if you, if something is misclassified, and especially as it relates to the clinical practice guidelines, then I think we need to sort of reconsider what is the diagnostic criteria in relationship to the natural phases of chronic hepatitis B. So look, I did mention in my defense to the comments made that if you actually do sequence patients who are so-called, or individuals, because they're not patients, individuals in the immune tolerant phase, that the hepatologists and the general physicians can feel comfortable about, well, if the virus is drifting and mutating, then it's not really immune tolerant, is it? It's got to be something else. And just on population-based sequencing, the cohort that Henry referred to, and by next-gen sequencing, 35% have already changed in the BCP and the Prius1, Prius2. So over a third of the patients have already evolved. So just in a few minutes before I start my presentation, I thought I'd throw out a new classification, a viropathological one. To me, that makes more sense. So I would call the immune tolerant a high replication, no inflammation, if you like, a virostasis phase. Now that, to me, that says something. Virostasis, the virus is not drifting, the virus is not doing anything, there's no inflammation, high replication but no evolution. I could probably accept no treatment. But if you're in a replication phase, E antigen positive and inflammation, it's a high state of viral evolution, then yeah, something's going on, something needs to happen, because if the virus is evolving, then it's ducking and weaving, and it's going to be doing something bad to your liver. Now, in terms of latent, low or no replication, no inflammation, low viral evolution. I could accept there that would be a non-intervention phase. Now, the word reactivation really does sort of stick in my throat. I don't like it. Replication, E antigen negative and inflammation, your high viral evolution again. Viral lows can be sort of misleading really, because the viral loads are said, they're lower in E antigen negative chronic hepatitis B, but the mutation rate is an order of magnitude higher. So I think that again tells me the virus is ducking and weaving, probably much higher than it was in the immune elimination or the immune clearance phase. This is certainly another phase that you would want to treat. Now, a very controversial phase that was introduced by Massimo Livero, some data from Giovanni Raimondi's group about occult. I think occult's a very important phase that we need to understand in more detail. To me, occult hepatitis B represents DNA, your surface antigen negative but DNA positive. You're almost there but not quite. You could call it immune control. But really the phase that we're trying to get to is, are we there yet? Are you cured? That's DNA cleared and anti-HBs is made. So your surface antigen negative, surface antigen negative, HBV DNA negative, and anti-HBs positive. So there, Ray would happily call that a dead virus. All right, so I throw that to the hepatologists. It's really time to come into the 21st century and reconsider some virology in the classification of chronic hepatitis B. Okay, so I could probably hang up now. But I think he'd be very keen for me to stop. Okay. The organisers have asked me to give a talk on how to achieve a cure, and a lot of my presentation has been covered by previous speakers, so I'll really just give you the virologist overview and briefly discuss treatment challenges, virological barriers, and future perspectives. And I think, again, most of this has been covered, that if we do use low genetic barrier drugs like lamivudine, then resistance is a serious issue. The long-term therapy compliance is another issue. And unfortunately, as we've heard, has little effect on surface antigen. I think a very telling clinical observation has been made for many times in case reports about rituximab therapy in persons who are so-called cured, cleared HBsAg, DNA negative and anti-HBs positive, they still readily reactivate to the tune of 20 to 30%. And I think as we see more and more rituximab being used for more and more clinical indications, we're going to have this challenge more and more. And really, pegylated interferon has plenty of toxicity. So since I'm into terminology and reclassification, I think it's good for us to know that we're all on the same page with our phraseology. Eradication was used in the context of the smallpox program. It equates to driving a virus to extinction from the earth, like in the case of smallpox through vaccination. Cure equates to eliminating the virus from the infected host, like has been successfully done now with hepatitis C. So what does that mean for hepatitis B? Well, I think, yes, we can eradicate it eventually through the global vaccination program, but that'll be in my children's time. Still, even with the most successful immunisation programs, we're still left in high prevalence areas with a background paediatric rate of 1 to 3%, depending on whether or not HBig is used and depending on how good the vaccine serotype mismatch or not is related to the circulating virus in that particular population. And I think, can it be cured? Well, after today's symposium, and hopefully the answer can be probably. So if we did achieve a cure, what would it actually look like? What would the hepatologists and the virologists finally actually agree on? And in the blood, we would obviously have DNA negativity, surface antigen negativity, and anti-HBs positive. I think there'll be a lot of discussion about what we accept will be in the liver. So we obviously don't want to have any cccDNA measurable by any assay. We don't want to have any relaxed circle or double-stranded linear DNA, because that would certainly imply active replication. I think an important marker immunohistochemically would be core antigen negative staining, either cytoplasmic or in the nucleus. But I would accept some occasional surface antigen positivity, because that would reflect integration from translation and transcription from integrated HBV DNA sequences. So what are the barriers that really the virologist sees in terms of curing hepatitis B? We've talked about this cccDNA, and I'll come back to that in a minute. We are restricted by the limited number of enzymatic targets. We've only got a priming reaction, a reverse transcriptase reaction, and a RNase H activity to deal with. So the strategies that we've looked at over the years to sort of overcome some of these barriers has been programs around depleting or silencing the mini chromosome, improving the potency of our inhibitors, broadening our targets, and of course, when all else fails, put it all together and hope for the best. But I'll actually show you some points that will hopefully clarify that just putting everything together is not necessarily going to be a good thing. this is a really nice review just published recently in Futures Virology, which sort of goes through the sort of key steps in the life cycle that I'll cover now. So as Harry's already pointed out, that the receptor's been discovered, the NTCP, sodium taurocholate co-processing protein, and this is a receptor that the hepatitis B virus seems to use a lot. And then from there, it goes, the genomic DNA is converted to a mini chromosome, which we'll look at in a minute. And then so a target will be to sort of block attachment through some mechanism, an entry inhibitor, block conversion and processing of the cccDNA, then block transcription, early and late mRNA through molecular-based therapy, and then look at the assembly reactions that the viruses do so well in terms of the nucleocapsid and the virion. I won't look at multiple rounds of reinfection. Now, just a word about cccDNA. I really hate that phrase because it was actually coined by a group of another set of health professionals who really have no regard for political correctness. It's molecular biologists. Now, cccDNA is physiologically irrelevant. It's never found in the cell. cccDNA can't be found in the cell because A, it's toxic, and B, as soon as any form of naked DNA is entered into a cell, it's rapidly packaged and processed and protected in transcriptional-based factors. So we never see cccDNA inside the liver. We only see cccDNA when we do a liver biopsy, pull it out and do a DNA extraction, then you have cccDNA. What you really see in the liver is a mini chromosome. And why I don't like cccDNA is because it conveys that it's a single substrate, whereas in actual fact it's a mini chromosome existing in a heterogeneous number of transcriptional states. And what John Newbold and I showed many years ago, actually, when I was a young virologist, was that it consists of this heterogeneous group of beads on a string, a full complement of mini chromosomes shown for you, of nucleosomes shown for you here, what we call compacted structures. And these are what we show with the low replication phenotype. Then you have an open structure, half chromatinized typically, both in human and in animal models. And these half chromatinized are presumably the transcriptional active, the high replication phenotype. And I'll come back to these in a minute in terms of actually inhibitors. So as we saw in the HIV from what Daria told us, is that the similar sorts of thoughts have gone into silencing or affecting the viral mini chromosome. And so how we do that is because it's a mini chromosome, then chromatin modeling enzymes like histone acetylases that notoriously are very transcriptionally active. If you inhibit those, it's going to sort of silence the mini chromosome. Whereas if you take histone deacetylases, which are actually involved in downregulating or silencing the mini chromosome, then you're going to activate the HPV replication. So the conclusion and what Massimo Livero and Giovanni Raimondi's group have shown is that the acetylation and the methylation status of the viral mini chromosome, mainly on H3, H4, is really what regulates the virus on a day-by-day basis and over the natural history of chronic hepatitis B. And that's reflected in the HPV viral load. Now, we've already got clinical validation of that through wider spread use of the histone deacetylase inhibitor group, and we've seen evidences of both not just hepatitis B, but also papillomavirus reactivation in patients, cancer patients who have gone on to these inhibitors. So yes, if you do use drugs that modify the chromatin, then you're going to sort of see effects on the hepatitis B virus. So what I've tried to put together here is two long-standing studies separated by 20 years, really. This is the science paper that Harry referred to, which shows that the lymphotoxin B and also that Massimo Livero had shown with interferon alpha activates ApoBec3A and ApoBec3B, and presumably through a postulated mechanism of interaction with the nucleocapsid or the core protein of HPV, which is intimately associated with the mini chromosome, end up in the nucleus and actually deaminate the mini chromosome. Now, the only mini chromosomes that ApoBec's going to deaminate and actually cause lysis of is this group of mini chromosomes here. And what this is a analysis of the mini chromosomal or the cccDNA. I can use that phrase in this context. You can see here the different topoisomers of the mini chromosome of HBV. The half chromatinized ones were the B1, B2 I showed you in the earlier slide, and these are the ones that are vulnerable to ApoBec 3A and 3B. The fully chromatinized ones shown for you here, 18, 19 and 20, they will not be affected by ApoBec 3B. And that's what these workers found, was that when you use these sorts of cytokines to stimulate this sort of mini chromosomal degradation, they only got a 50% reduction. And why is that? Because half the mini chromosomes are transcriptionally active, the other half are transcriptionally silent. Now, we are actually making some progress. We've got assays now, cell culture systems, for looking at inhibiting mini chromosomes. Tim Block's group under Haitao Gao have shown that the disubstituted sulfonamide compounds do seem to have some selectivity for what they called cccDNA. But they need to prove that it's not actually at the level of the core protein, and that it's actually at the level in which particular form of the mini chromosome is actually affected by these sorts of compounds. Now, I'll come back to the mini chromosome in a minute, but I'll go on to the next target. We've heard a lot about TAF, and as I said, we've got a limited number of targets, so obviously improving the potency of drugs like tenofovir into tenofovir alafenamide is a good step in that right direction. And you can sort of see here in terms of HIV activity how we've got orders of magnitude increase as we go from tenofovir to its diisoproxyl intermediate, the tenofovir here, or prodrug, and then now TAF, the next one on the block. So I think that as the drug gets approved and rolls out, it'll be interesting to look at how the impact of improved potency works. All right, so we've gone through the mini chromosome. How about the next new target on the block? Receptor blockade and entry through the sodium taurocholate co-transporter new receptor structure inhibits both human hepatitis B and hepatitis D. We're all very excited to be looking forward to the results that are going to be presented hopefully at AASLD on the Delta virus story, or hopefully soon thereafter. Now, we're through, I guess, two independent approaches. The Chinese hunted for the receptor using knowledge that the Germans had discovered through Stefan Urban's activities by reverse competition studies, that he argued that the key ingredient for receptor binding for hepatitis B virus would be in the pre-S1 protein, and he took a myristylated form of that from amino acids 2 to 48, which included this 10-mer sequence here, which, as it turned out, is the critical region involving in binding to the NTCP. And this is why Mercludex blocks re-entry and entry of hepatitis B virus back into the hepatocyte. And so shown for you here are some mouse model studies done with Maura Danby's group, showing how effective the Mercludex B is in certainly inhibiting HBV and Delta replication. And as you've heard today, Mercludex B, the first entry inhibitor in hepatitis B terms, and Delta is completing phase 2 at the moment. Now, I'll just finish off with assembly inhibitors. The viral targets here for viral assembly include nuclear capsid assembly. I'll include assembly of the viral mini chromosome, and I'll include assembly of the virus itself. So in terms of nuclear capsid assembly, there are 3 steps that are considered and actually actively investigated. We've got the packaging reaction, reverse transcription, and there will be or could be an effect of these compounds on the actual e-antigen status secretion as well. Now, reverse transcription in hepatitis B is a mighty complicated process. It involves 2 primer translocations and 3 template switches. And so all this has to be orchestrated, unlike HIV, in the context of the viral nuclear capsid. And that's why the virus replicates so dependently on nuclear capsid. So a number of chemical compounds have been trialled and identified in screens. The phenylpropanamide inhibitors from Avid Laboratories that have now ended up in the Gilead stable. The BHAPs that you heard Ray talk about with the Bayer compounds, and other compounds from Novira and companies that are targeting the core protein. And the reason why these compounds are being made is that we have a crystal structure for the hepatitis B core protein. Now, this is Jordan Feld's first paper. I thought I'd show it for you, Jordan. And this is when he was out of my lab for a little bit of a sunny sabbatical. And the phenylpropanamide 8061, 8130 compounds certainly inhibit HBV, but not as well as, say, lamivudine, But when dissected out in terms of the context of the replication cycle, you can see here that AT130 has no effect on total RNA replication, but selectively blocks the RNA inside the capsids. And he went on to show that this RNA was exquisitely sensitive to the packaging reaction through the core RNA interaction, was exquisitely sensitive to the phenylpropanamide compounds AT130, shown for you here by almost no level, complete inhibition of the packaging reaction. The heteroaryl dihydropyrimidines, also they're more core busters. They're not selective like the phenylpropanamides, which block packaging. These are more actually just destroying or blasting up or blasting away the nuclear capsids, and you get a classic non-nucleoside inhibition profile in terms of dose-response curve. Now, as I said, I'll come back to the mini-chromosome at this stage. Thomas Bock showed very elegantly in the presence of core that the core protein actually specifically associates with the viral DNA that made it in the nucleus of the cell. And it actually behaves like a histone in its own right. The core, when it binds to the mini-chromosome, introduces a positive supertwist, or the equivalent of a supertwist at 20 base pairs. So it increased the linkage number from 15 to 16. And here's some of Thomas's photos showing very nicely the core protein associating with the viral mini-chromosome. It's a physical, direct association, unlike the association of X. So that has to be considered. So therefore, if you're thinking, well, if I'm targeting the mini-chromosome and I'm targeting the core protein, will it be a synergistic or an antagonistic effect that I'll actually end up achieving? So I think Thomas's work is worthwhile recalling the fact that not all combinations will necessarily be additive or synergistic. So, and this the point I wanted to make with this slide is that ApoBec has at least two groups of activities, deamination dependent, obviously through its activity, and a deamination independent through its apparent core binding through the RNA pre-genome epsilon interaction. And so that was just to sort of remind everybody that, yeah, if we knock the core protein out, will we also affect the ability of ApoBec to actually end up in the nucleus and do the right things in terms of its cytidine deamination? All right, so the last group of targets that we'll look at is virion assembly. Obviously, the major focus at the moment is the hepatitis B surface antigen production. And really, many, many years ago, at the height of molecular biology, we had a very large number of molecular-based therapies that were mainly targeting the viral messenger RNA, and obviously through antisense and silencing, and you heard about a lot of these today. And really, they all fell over because delivery became a huge problem. And now that's all changed. Oh, before I do that, so I'll just take one of these as an example, and the one that's actually in the clinic. Arrowhead in phase 2 with their ARC 520. And the cell has a particular way of dealing with double-stranded RNA through the RNA interferon pathway. And by using the DICER RISC complex, double-stranded RNA molecules are chopped up because they're pretty toxic. They're cleaved and processed to the RISC and then generated into single-stranded RNA. And that has been exploited to do selective gene silencing in terms of targeting genes, and probably how the microRNA apparatus works as well. So it's now possible to deliver synthetic siRNAs to basically go, if you have the 18 to 21 nucleotide length set up for you, goes through RISC, and you can do selective gene targeting. Now, as I said, the big problem with all the molecular-based therapies in the past has been delivery. And Arrowhead have got this dynamic polyconjugate technology, which consists of a sort of amphipathic peptide. They're able to target that particular liposomal structure to make it as liver-specific as they can through N-acetylgalactosamine, and package the siRNA in this particular structure to be able to deliver it effectively to the liver. And they used, here's the hepatitis B genome linearized across all of these various RNA molecules. And you can see here the region where there is 4 RNA transcripts, the pre-core, pre-genomic, the envelope, as well as the X. This is where they chose 2 silencing RNAs and to deliver into their DPC technology. hoping to get the most effective. So they're not just going to block surface antigen or pre-S1, they're going to block core, they're going to block X, they're going to block E as well. And so where that all fits in, this is where you're used to where the nucleotide analogues block. This here the reverse transcription. Now the molecular-based therapies are more targeted towards the RNA, and so what you end up with is RNA blockage, mainly through the mechanism, through the silencing reaction we've just gone through. And their hypothesis, which is yet to be proven, is that by blocking S and blocking E, that you'll actually revive the adaptive and the innate immune responses and generate a functional cure. So they have to achieve knockdown of all of the RNA replicative intermediates and then wait to see whether or not that's been effective in terms of immunological recovery. In the transgenic mouse, the ARC520 is the double RNAi that I just showed you before. There's the control animals, and here all of the core antigen expression in the transgenic mouse was rendered unstainable. And then Robert Lanford has presented the chimpanzee data, E antigen positive chimp. They've got 2 to 3 log knockdown in DNA, E antigen, and in S in terms of that one particular ARC520 formulation, 2 milligram per kilogram dose here, followed by a 3 milligram per kilogram dose here. So the next steps are poised for, if we can do that for S antigen, we're blocking for core, can we actually block for virus? We now know that there are certain interactions between the L protein and the core protein. Again, through peptides, we can block that very effectively, but again, delivery has been the problem. So if the DPC technology works, ends up in the liver, then lots of these molecular-based therapies can be applied and translated over to treating hepatitis B. So we've sort of covered attachment, mini chromosome generation and processing, viral early translation and transcription, and viral assembly and release. So you can see that the pipeline is good, as all this slide shows. Lots of people doing lots of things in hepatitis B, as Professor Shanazi has pointed out. And so I pinched this slide from Bill Delaney, I think. And what would a hepatitis B curative regimen look like? Well, it needs a potent nucleoside analog. I should have said a mini chromosome inhibitor, and immune activator, and excess surface antigen producer. But we really have to then titrate these as best we can in order to effect a virological cure. So I think it's very exciting. It's great to be in B, and the goalpost is shifting. I think Pietro's is happy with DNA suppression. I still think the goal should be S antigen clearance. And so I think we're now sort of pushing hard for that, and lots of new agents are coming. I think the NTCP cell line availability will really improve the lead compounds, and I think now that we've got liver targeting with these DPC type approaches, then I can sort of feel palpable optimism in the hepatological community. Thank you.
Well, thank you very much for the invitation to present this talk on the immunological perspective. I do hope everybody has some coffee left out there, because we are going to talk some pretty serious immunology. But it's an exciting time, because I'm going to show you a lot of studies here that are really only published in the last 2 years. And I'm going to go through the slides pretty quickly because there's so much to cover, but we actually can find most of these references in a recent review that Antonio Bertoletti and I published. So to get started, in terms of an immunological perspective, we need to understand what you need for immunological control of hepatitis B virus. And to be honest, in general, this is not much different than you see in any other normal acute viral infection. You need T cells that can recognize an infected target, and most importantly, produce cytokines, antiviral cytokines like interferon gamma, which is actually responsible for a majority of the non-cytopathic clearance of the virus. And then, of course, you have antibodies that neutralize the virus. And the big difference here is it takes 2 to 3 months to clear acute hep B versus another acute viral infection. So when you look in the context of a chronic infection, you don't see this multi-specific T cell response effect. It's very weak. We have very few antibodies or ineffective antibody responses, and the T cells, if they are present, are actually prone to apoptosis, or they co-express a number of inhibitory receptors like PD-1, CTLA-4, and TIM-3. So this is actually quite easy to quantify experimentally as well. So if we do an ELISpot, for instance, using overlapping peptides covering the hepatitis B genome, and you look in a resolved patient, each one of these black bars represents a T cell response in a resolved donor and a resolved patient. You look at this same response in a chronically infected patient, and about 80% of the time ex vivo, this is what you'll see, is virtually non-existent. So I think there's two very broad and very general reasons that you see this level of exhaustion of this immune suppression in chronic hepatitis B. One is the fact that it's a hepatotropic virus, and we're quite aware that the liver is generally a very tolerizing organ. You have high IL-10, TGF-beta, PD-L1, enzymes that degrade arginase and tryptophan, which are all very suppressive for T cell responses. Coupled with low MHC on the hepatocytes as well as suppressive dendritic cells, makes it difficult under resting conditions to stimulate an effective immune response. Coupled with this hepatotropic infection is the high antigen burden that we see in the hepatitis B patients. And this is one of the things that fascinate me the most probably about the virus, is that it's incredibly small, makes only 4 proteins, but its synthetic effort is really phenomenal. I mean, the level of viremia in these patients is often 10 to the 9, can be 10 to the 9, 10 to the 10, and I've worked with some of these patients where in their serum we can see up to 1 milligram per mil of viral antigens. So there's really a massive antigen burden in these patients. So then the question in terms of immunology is, how can you actually restore a virus-specific T cell response that's been exhausted from years of exposure to an inhibitory environment like the liver, and coupled with the amount of antigen that's present in the circulation or in the body? So your first thought, obviously, is therapeutic vaccine, or at least for an immunologist, is therapeutic vaccines. And Dr. Jansen attested this before. We've been trying now for at least 15 years, and I could go through each of these individually, such as recombinant antigen-based vaccines like the prophylactic vaccines, or going and adding a TLR agonist to immunodominant peptides, DNA vaccination, or immune complexes with alum. But the ultimate response is that after vaccine therapy, we see a transient response. There might be a little bit of a boost in the T cell proliferative response. There might be a slight reduction in the viral DNA for a little bit, but ultimately there's not a durable response. You don't get any good CD8 T cells, which are really responsible for the clearance. And there's very little S antigen loss. So there has been some advancement, though, in the vaccine field, and so in a lot of this, and I'm going to come back another study later, is through the Rogendorf group using their chronic woodchuck hepatitis model, and a new approach, or the most updated approach for inducing T cell vaccines, this combination of antiviral therapy with a prime boost regimen that incorporates adenoviral vectors with a DNA prime boost strategy, and this has been extremely effective in animal models like the mouse and the woodchuck at really inducing a good T cell response. The caveat to this is they have to hit these animals with many, many doses of the vaccine. So you can see they give, it's hard to read actually, I'm sorry about that, but it's 3 doses of a DNA vaccine alternated by 4 doses of an adenoviral vaccine, and then coming back with 2 DNA vaccines, all in the context of antiviral therapy. So giving this kind of aggressive regimen, you do start to see this restoration of a CD4 T cell response about halfway through the vaccine regimen. But in fact, the CD8 T cell responses are much more delayed. So they come up here at about the end after you give the last 2 boosts of DNA vaccine. So what does this look like in terms of a virological response or an immune response to clear the virus from these chronically infected woodchucks? Well, in this woodchuck, it looks fantastic. So you start off at the beginning of therapy, entecavir starts to reduce the viral load, and upon starting the immunization schedule, you see a reduction, almost an elimination of S antigen, and then the woodchucks become anti-S positive. So the caveat, though, to this one is that the S antigen here in this woodchuck is extraordinarily low. It's 5 micrograms per mil. And when they actually looked at the other animals in this group, there was only 4 in the whole study, but when they looked at the other animals that had a much higher S antigen, you can see that it does come down and DNA goes away, but after stopping and after the end of the vaccine therapy, even this very aggressive approach is not necessarily able to end up with stable control over the virus. So it does look like there is some benefit because you get the reduction in the set point for the S antigen, but it's still not be able to clear. And I'm going to give you my hypothesis as to why this is probably the case. You may not agree, but I think this is the case because when you talk about vaccines, you're talking about a needle, a shot in the deltoid or in the arm, and you're giving a very local injection. So in the case of recombinant antigens, a lot of times it's 10 to 50 micrograms of antigen. If you come back to what I said in the introduction about the synthetic effort of the virus, they have high viral load, and it's not unusual to see a patient with 20,000 units of S antigen in the serum. So if you just do some quick numbers based on this, you have 5,000 milliliters of blood, 100 micrograms per mil of antigen in the blood. That's 500 milligrams of S antigen in the patient. So a vaccine with 50 micrograms is equal to half a milliliter of the blood. Why should the immune system look at what's going on in the shoulder when it's overwhelmed with antigen in the rest of the body? So that presents an even bigger question. How do you actually deliver a stimulation to break this virus-specific T-cell exhaustion? And so what I think is you really have to think on a larger scale and reshape the entire immunological environment. Think systemic versus the local injection of the vaccine, and try to exploit the natural immune mechanisms and also take advantage of the viral life cycle, because the virus is giving us plenty of antigen to use. There's no reason we have to put more antigen in to get an immune response. Plus, the antigen you're putting in is not matched to the virus that's in the patient, so you have issues of viral diversity. So how do we do that? Well, I think there's actually hints from some of those studies I showed you in the previous 2 slides. So this vaccine study with the immune complexes in alum, the responses in this group were to the vaccine were no better than alum alone. But if you look, I don't know if you can see it, but these patients with alum alone, 20% of them seroconverted E antigen. So even the adjuvant alone had an effect here. It was actually a little bit better than the vaccine, which was 14%. And then here in this DNA adenoviral prime boost study from the Rogendorf lab, in their woodchuck hepatitis virus mouse model, they were hitting this mouse with a core antigen vaccine over and over, but then they saw a boost in the anti-S antibodies, suggesting that this kind of adjuvant effect has could have some kind of immunological effect overall, not specific to the vaccine. So then one of the things I'm particularly interested in, and a lot of studies that are coming out to support this, is can an adjuvant or an immunotherapy actually rescue the HPV-specific T-cell responses? And in terms of adjuvant therapy, I think the one that we're probably all most familiar with right now is this, the Gilead compound, this GS9620. And the concept here, this is a TLR7 agonist, and the original idea is it stimulates interferon alpha production, which then goes on to have its antiviral effect. And so the good thing about this is, TLR7 is very immunostimulatory. Maybe the problem with interferon alpha is we know it's not necessarily curative. There's good studies now to show that it does not restore T-cell responses, and then obviously it has its side effects. But interferon alpha is obviously not the only cytokine that's going to be produced in response to TLR7, so there might still be some adjuvant effects we haven't really seen, or at least I haven't seen data on the T-cell responses of HPV-specific T-cell responses so far after administration of this compound. And then we published a study just this year looking at TLR8 specifically, and TLR8 stimulation of intrahepatic monocytes. And what we found was that when we stimulated intrahepatic monocytes with this TLR8 agonist, they produced loads of IL-12 and IL-18, and this worked on NK cells and mate cells, this mucosal-associated invariant T cells. This is an NK T cell in the human liver, and it stimulated these cells to produce massive amounts of interferon gamma. And these 2 lymphocyte subsets probably compose 40 to 50% of your intrahepatic lymphocytes. Interferon gamma is critical for the non-cytopathic clearance of hepatitis B. So, using the right TLR adjuvants, you might get the right antiviral cytokines to get hepatitis B clearance. This was not done in the context of hepatitis B, but it's an extrapolation from the data. And then there was another study just published last year from Percy Nola's lab looking also at the activation of intrahepatic monocytes, but in its case, in the context of TLR9 stimulation. And when they stimulated the intrahepatic monocytes with TLR9, they found that they produced TNF-alpha and recruited T cells to the liver. And they formed these aggregates, and these aggregates facilitated intrahepatic T cell proliferation. And so you got this massive boost in the HPV, or in the antigen-specific T cells in the liver. And so when they combined this TLR9 immunotherapy with a DNA vaccination in chronic hepatitis B infected mice, what they found was that there was a much better response in response to this combination vaccine and adjuvant therapy, vaccine and we'll call it adjuvant therapy, compared to either the adjuvant alone or the vaccine alone. So suggesting that this modulation is having a beneficial effect as well. But there's also immunotherapy, and this is slightly different because those were TLR agonists stimulating antiviral inflammatory cytokines. But there was also a paper published just last year from Masanori Isagawa from Frank Jazari's group at the time, where they targeted myeloid dendritic cells, these cells that are very important for presenting antigen and initiating the immune response. And instead of giving them a TLR agonist, what they did was target a co-stimulatory molecule, CD40, on these dendritic cells, which upregulates their ability to process and present antigen. And in their HBV transgenic mice model, when they gave them T cells without this adjuvant therapy or without this immunotherapy, the T cell response was weak, hardly ever produced any cytokines at all. However, when they gave this anti-CD40 therapy, you can see this massive boost in the CD8 T cells in the liver. A lot of these cells produced a lot more interferon gamma, and you can see in the blot over here that it almost completely eliminated HBV DNA from the liver in the context of this adjuvant therapy. And so another thing that we've been interested in is along these lines as well, thinking about the antigen burden in the serum of the patients, we wanted to see if any of the monocytes of the dendritic cells in the patients were actually internalizing and using this antigen. And what we found was that this classical population of monocytes, the CD14 monocytes in the blood of hepatitis patients, were loaded with the S antigen. And these are consequently the cells that become dendritic cells and present antigen, and they are the same cells that we use for DC vaccines. And so we simply asked, if we converted these monocytes to dendritic cells, can they present that antigen that they accumulated in the circulation of the patients? And seen here, we saw in 8 out of the 9 patients, this was the case. And what we did, we went on to differentiate these cells to dendritic cells, mix them back with their autologous PBMCs, and saw expansion of autologous T cells, suggesting that there is a therapeutic effect. So that's well and good if there's a T cell response to boost, but I think in many cases, these T cell responses can even be deleted in this really, really high antigen load patients. And so what do we do then? And in that case, there's also an arm of research going on that is much more experimental in nature, but you make a T cell response. You engineer your T cell response that you're interested in. And one way to do that is through a chimeric antigen receptor. And basically, this is an antibody that you modify, clone the DNA, put it into a retroviral vector, and then drop it into the T cells of a chronically infected patient. It's easy to detect this, so when you put it in, now you have T cells that will recognize the viral protein, in this case, the surface antigen. And these T cells will recognize the surface antigen on the surface of hepatocytes, and when they put them into HBV transgenic mice, they saw that these modified T cells reduced the HBV load in the transgenic mice. So this is another thing that we've been working on as well, but also in a slightly different context, in T cell receptor gene therapy. And what T cell receptor gene therapy is, here we actually identify T cells from resolved hepatitis B patients that we know recognize infected hepatocytes and can kill them. And so we clone the DNA that encodes for that receptor, and then it's the same process. You put it into a viral vector, and then you introduce it into the lymphocytes of a chronic hepatitis B patient. And you can see very clearly, if you don't introduce the receptor, there's no positive population that are HPV specific. If you do, we see a nice population of T cells ready to respond to hepatitis B infected cells. And we showed that these cells were functional in an in vivo mouse model where we put a tumor in, let the tumor grow, and then we transferred in these genetically modified T cells. In the control, you can see the tumors here, and then a dose-dependent reduction in the tumor volume in these mice. So suggesting that these T cells can target tumors expressing HBV antigens, which is an important concept because this happens a lot of time in HBV-related HCC. And in fact, through a fantastic collaboration with colleagues in London and colleagues in Pisa, we're able to use this exact therapy in a compassionate care use basis on a patient that developed HBV-related HCC. So just go quickly through the medical history here. And so the patient was diagnosed with chronic hepatitis B, developed liver cirrhosis, and then 2001 developed HCC, underwent transplant, was fine for 5 years, and then detected HBs antigen in the circulation. So after that, they found that the patient had developed metastases, but these metastases were actually from the original liver. They weren't from the new transplanted liver. And so they tried to surgically treat this, but ultimately we got compassionate care use, the possibility to use our genetically modified T cells, because what we could do with our T cells is we could make T cells that specifically recognize the metastases, but not the liver of the patient. So it minimizes any risk to causing liver failure in this case. And so we did this, and so this is a history of the S antigen here from the patient, looking from 2011 up till 2012. You can see a dip upon surgery and resection, but it kind of gradually increases. And at this point, we infused the genetically modified T cells, and within 30 days, this went from about 3,500 units per mil down to about 500 units per mil of the S antigen. And so this drop in S antigen correlated with an increase in the frequency of our T cells in the peripheral blood of this patient, as well as a slight increase in ALT, presumably coming from these tumor cells that are being lysed. So this is, unfortunately, it wasn't curative in this case, and the patient did succumb to a cerebral metastases, but there was no adverse effects from this adoptive cell therapy. And I think these are encouraging results, even though it is one compassionate care use basis. But the fact that we did see some biochemical evidence that these cells were effective. So just going to wrap up quickly, and unfortunately I didn't get to put everything in here because, and some people have talked about these use of checkpoint blockades in previous talks, and I think there is a lot of potential here, especially, there's a lot of great in vitro data using the blood from chronically hepatitis B virus infected patients. And then back again from the Rogendorf lab, they did this vaccination protocol with chronically infected woodchucks, either with combination therapy or combination therapy plus anti-PD-L1 blocking. And in the blue line here, you can see the anti-PD-L1 effect. And really, it seems, at least at the end of their follow-up, that the DNA is quite low. They didn't necessarily clear S antigen in this case, but it seems like it's more than effective than the other vaccine regimens they use. So there might be some hope here as well. And certainly there's some good data coming from the cancer field related to this as well. Then one last thing that I'd like to point out, just because it's one of those things that, I don't know, I've always thought about it but never done it, and nobody knows anything about the B cells. I put this black box there because that's what we know. It's very little study that's actually ongoing about the B cells. And we know it's important because if you use rituximab in these chronically infected patients, they even develop reactivation. So it doesn't even have to be anti-core positive healthy donors. So I'll just plug that, but no plans. Anyway, so just to summarize then quickly, I think adjuvant therapy has the potential if it's used right, because it's activating these toll-like receptors, and we're altering the immune environment in the liver. And this, in terms of, this facilitates a T-cell response and T-cell expansion in the liver where we need it. These immunotherapy approaches, this one is, I think these are really interesting because we're using the antigen that's in the patient. So we're personalizing the antigenic repertoire for each individual by giving a general immunomodulatory combination. And if we can stimulate T-cell expansion, antiviral cytokine production, I think there's a real benefit there. And then, of course, there's more experimental therapies like the gene therapy, but they're in development, and things are moving in this field and moving fast in the cancer field. And they've been effective in animal models. They appeared effective in our patient therapy, but there's a lot more data that are needed. So I will stop there, and I appreciate everybody's attention. Thank you.
I saw the program, I thought, whoa, what shall I tell you about a clinical perspective after all these talks? We had the introduction by Ray, we had the talks in the first session, all the clinical data have been mentioned, we had a long discussion, we had now the virology and the immunology, so all comments actually were made. I therefore thought to summarize some of the ideas maybe and to remind ourselves what we're talking about. And when I talk from now on cure of hepatitis B, I do not mean control of viral replication. I really mean seroconversion, which is some kind of functional cure. And we have some tools that this developed already. Genotype A infection, interferon. For 10 years, we raised the arguments that this might be a goal. But in real life, we have to say, who cares about cure? because the winner in clinical practice is clearly HBV DNA suppression, even though only considered as a third endpoint, but this is what we're all talking about. So S antigen, does it really matter for us in clinical practice? And also these arguments have been raised. I think we have already the perfect therapy. Do we really need curative therapies? We have HBV polymerase inhibitors that really work in every patient. You've seen the data on fibrosis regression. These drugs are extremely safe. We didn't talk about this. Even the issue of tenofovir and kidney failure, which is an issue in HIV infection, in real life, in HBV, this is really a very, very minor problem. And it has been shown that these drugs prevent clinical endpoints, as pointed out by Harry, and even HBS loss occur in some of these patients. So everything is fine. These S losses are rare, but they are important, and they also have been linked to a reduction in the incidence of HCCs, and I think Harry showed this slide before, or Henry, so I think this is fine. However, do we need these curative therapies? And we raised the arguments for yes. The issues on lifelong therapies, costs have been made, endpoints develop in patients who are S positive but suppressed, so there is still an issue that they may develop cancer. My first point, however, is S loss really the appropriate surrogate endpoint to predict that these events occur less often? And I will come back to this paper in a minute from Markus Kornberg, where we investigated S antigen kinetics during NUC therapies. But I saw 2 weeks ago one of these patients. This trial was published 2011, and this patient actually lost, after 12 years of lamivudine therapy, initially E antigen negative, he lost his S antigen. Very nice, 70-year-old man. But you know why I saw him? Because he developed cancer 2 years after S loss in this case. And therefore I come back to the issue what we discussed before of age, when to start. Didn't we simply start too late in this patient? There's also evidence from the spontaneous clearance of S antigen that it's better to lose, obviously, S antigen before the age of 50. This might be one cutoff. We should treat earlier, but if we start these approaches of curative therapies too late, in the 50, 60, 70-year-old persons, we might be simply too late. And again, the issue of number needed to treat from the clinical perspective might be challenging in this concept. And then there's the other argument, and this has been raised in one of the questions. Obviously, there's one scenario where we definitely need curative therapy for hepatitis B, and this is Delta. Patients are dying from hepatitis delta. The delta virus is using S antigen for transmission, and there's a lot of data that this is the most aggressive form of chronic viral hepatitis. Data from Greece published last year again showing that HDV positive patients have a more severe outcome than mono-infected patients, and you can group delta patients with different risk profiles. This is data from our group where we have some patients with mild courses called BIA, but then we also have patients with a more aggressive course, and if you apply this clinical score to other centers, it does not only work for us in Hanover, it also works quite well in Barcelona and in another German center. So there are patients with a very severe course, and our problem is that interferon, as discussed, has very limited efficacy. We have performed quite some trials together with Cianididine from Ankara and also with centers from Greece and Romania. And the key findings from our trials published in 2011 and presented this year at EASL were that 1 year of interferon may induce in about 1 out of 4 patients an HDV RNA suppression 24 weeks after the end of therapies. If you extend treatment to 2 years or if you combine with tenofovir, you really do not increase response rates significantly. And maybe most importantly, interferon is not only transiently effective in a limited number of patients, there's also no cure, because in the long term, there is no sustained virological response, as even if you are negative 24 weeks after interferon treatment, you may have late relapses, which we published this year in hepatology. So at least half of those patients with a previously called SVR actually did not have a sustained virological response and have late relapses. So there's really a need, in particular for Delta, to induce something, and this is literally cure from hepatitis B virus infection should be the cure for Delta. I've been asked to talk about the clinical perspective, and when I see my patients, obviously, it's nice to hear about core protein assembly mediators or genetic approaches, as pointed out by Rachinazi. I think we have to look what we have right now. And Harry briefly mentioned this. We have interferons. Can we combine this, yes or no? And what is actually the data to use interferon early? This is the standard treatment, 1 year. Few patients may lose S antigen. There might be the option to simply extend treatment, 2 years, maybe even longer. We may combine it from the very beginning. We may have a short interim combination. We may start with NUCs and then switch to the interferons, and we may do it even the other way around. So what is the data that has been actually published on all these different approaches? Different studies, different cohorts, and it's quite hard to summarize if a combination works or does not work. Different durations have been used, 16 weeks, 24 weeks, 48 weeks. Just some data published recently. Pietro published in Gut in E antigen negative patients, a trial comparing 1 year versus 2 years of therapy. Well, in some patients, there might be a small benefit, but really not justifying, and Pietro may correct me, to treat all patients now for 2 years. The question is how to identify these patients. There is data that will be presented, I think, on Monday or Tuesday at AASLD from the Gilead trial, the large trial with the combinations of tenofovir and interferon, and it's really a small increase. I think it was 8 in the abstracts. You can read 8% of S antigen loss if you do early combination versus 0% actually in patients treated with interferon alone. Then obviously it may differ in terms of the different NUCs. In our case, when we used the combination in Delta, we did not see an effect. And there I want to highlight, we've been talking about NUC therapy combining with something. I think we have to have a careful look what type of NUC. Different NUCs may differ. And for example, in our Delta experience, when we treated patients with interferon, either with adifovir or with tenofovir, we saw these surprising differences that in the first trial, we actually saw a better S antigen decline when combining interferon with adifovir, while we did not see any effect at all when combining this with tenofovir. So the question is, why is adifovir simply toxic, and by this inducing some kind of additional immune responses? Something to be investigated. And also, if you really look carefully in the data, combination tenofovir plus interferon versus entecavir plus interferon, there might be also differences. So it might not be that everything is simply the same. Then what's about this approach to use a short-term interferon course, maybe only 16 weeks? This is also one arm of the Gilead trial. And if I read the abstract correctly, there was absolutely no effect, only 1% of S loss. What's about starting with a NUC and then switching to interferon? There was no effect on E antigen loss in this trial from Asia presented at EASL. And actually, one of my favorite approaches, starting with interferon, then switching to NUCs, there's really no large controlled trial. At least from the clinical perspective, from single patients that we treated in this way, this might be something that may have been understudied. This was early interferon. What's about using interferon a little later? This is the approach, as Harry mentioned, of the paper that was published, I think, last week in Hepatology or went online. Then you may let's say, you stop both drugs after having added interferon. You may switch after long-term NUC to interferon. And also the discussion of low-level ongoing replication. Is there a role of combining NUCs in terms of S antigen levels? What is published here? And this is a trial which was presented at EASL and published last week. And this is also one paper, or one figure out of this paper. Harry Janssen showed you this data. But what I would like to highlight here, you have this decline in S antigen early during therapy, but then you have the stasis. There is no further decline here, and only a subgroup of patients in the end respond. It's also something which we have to understand. Why do we have this initial decline? Why not later on? Why is this not continuing? Do we need to extend interferon here? I think many issues to be investigated. What's about this approach? There is indeed an increased HBs antigen loss, presented also in an Asian trial at EASL. Here, there is some data also for this approach. If you switch, that you see some S decline, and the OST trial, which has been presented quite some times at different liver meetings, was published in the Journal of Hepatology, also showing a small increase in S antigen declines. But overall, we always talk about an increase of 10, 20%. It's never the magic bull that you can achieve something in 50, 60, 70%, which we actually what we want to see in the clinic. And what's about effects on S antigen levels or S losses with this combination? And this was mentioned earlier, and the trial by Henry was already mentioned that at least the number of S losses was not higher in this combination arm as compared to mono arms. Whether there are, let's say, distinct levels in S antigen quantitative levels remains to be determined. So overall, however, with all these approaches, we have to keep in mind that there are differences between E-positive and negative patients in these trials. And also, and this was mentioned by Adam, the role of quantitative S levels in the Roggendorf paper, for example. Quantitative S levels may be very important also in determining which patient is responding and less responding. So do we need new drugs after all what I have shown you? The combination with interferons will still need to be explored, but obviously with all these different approaches, not the magic bull. But stopping NUCs is already possible. And I was surprised not seeing this slide in all these talks before. The famous trial by, or study, or series of patients published by, from Greece by Stefanos Hatsianis, that indeed you can stop NUCs after quite some time, and some patients, half of them, have even an S loss. However, overall, in our clinical experience, this works only in a minority of patients, and the key thing is really that we need markers to identify patients which may benefit from these approaches. And different biomarkers have been explored, but I think we need much more work. One role could be, for example, IP10. And here I have to remind you, in hepatitis C, we have been used to data that high IP10 levels are bad. In hepatitis B, they are good. So a profound difference between the two viruses here. And if you have higher IP10 levels as a surrogate marker for immunity, interferon response, etc., then you have a higher chance to lose S antigen or experience an S antigen decline during NUC therapy. So what do I want from the magic drug X, which would, let's say, induce these cure of hepatitis B from the clinical perspective? And I gave already a name, and I really here want to highlight that if somebody is using this name, that I mentioned this here, so I name it Eradicate, not Eradicate. You can use it as an add-on therapy, you can switch it, or we may even use this drug as a first-line therapy. And for me, the question is, what are the musts for this drug? What do I want to see? And the most important thing, in the light of the fact that we have NUCs which are extremely safe, no side effects at all, any new therapy must be absolutely safe. I think we cannot accept and tolerate any significant side effect profile, which is a hard hurdle already for many drugs in development. I want to have finite therapy. We have definite and long-term therapies for many drugs. How long? Ideally 3 months, mostly a year from clinical perspective, which would be my profile for that one. And I want to see S losses, and which number? We see with interferon 10, 20%, and all this different mix and match of NUCs plus interferon. If this drug would develop, I would like to see at least 30% S loss, ideally much higher. But this is something which may be worth my continuing development. The question is really then, from the clinical perspective, do I need for a phase 2 trial S loss as an endpoint, or would be S antigen decline sufficient? And here again, we have to keep in mind the different kinetics of S antigen decline, and coming back to Adam's data or importance on the burden of viral load. We have some patients who have NUC therapies for a long time who will remain at the rather high level, and you have other patients who show a decline of S antigen levels during therapy. This has impact. And these declines are also different during the course of therapy. For example, if you would group patients under NUC therapy who have declines and you investigate the HBs antigen decline early during NUC therapy, you can see here that this does not really predict long-term S loss in these patients or S decline, because even those patients who do not show an early decline can experience later on an S loss. While if you group it in a different way, so once patients have become HPV DNA negative, then you can see here that those patients who have a decline here lose S in the long term, while this does not occur once S antigens are rather stable. For the magic drugs, Eradicate, which we want to use now in this context, it may make a difference if you use it here or if you use it here. So, for example, if you have a very potent new drug which induces a strong S antigen decline, it doesn't matter. But what if this effect is not that pronounced, and if you then need some time to become negative, and if I don't want to use my drug for more than a year or so, what do we do in this setting? Do we continue therapy? Is this clinically meaningful? What's about number needed to treat to prevent one HCC? I think these are really challenges for the clinical development. These were the musts to have. What's about the nice to haves? Obviously, the drugs should be easy to administer, ideally in an oral. And if I need to inject something in the year 2015 to a patient with hepatitis B who is happy with his NUCs, daily injections, is it justifiable? Once weekly, maybe, but obviously it's something which can be a hurdle. I want to have a drug. My Eradicate should be effective in all stages of HPV infection, including immunotolerant patients. I want to have a drug that works both in E-positive and in E-negative patients. And I want to have a drug that also may reduce the risk for HPV reactivation. We talked about this, rituximab, B cells, the effects. I want to have a drug which I can give to carriers, get rid of S antigen, induce anti-HBs, and then maybe safely to go for their treatment for vasculitis or lymphoma or whatever. In a best way, it should be a B drug, which I can use in pregnancy. We talked about pregnancy and to prevent transmission. This would be also nice. And ideally, it would be a drug which I can use in resource-limited setting also as a first-line therapy. And then for a clinical development should be interferon my comparator. And how to use it here, again, if it would be very potent, would be fine. I may have some slower declines, but I may also see, let's say, S antigen positivity after stopping the drug, and then it's a question, will this continue to decline? Will it be stable thereafter? And then the same question, what shall I do in this context? Is it clinically meaningful? Should I continue therapy? And lastly, what do I need furthermore from the clinical perspective? I think when we develop new therapies, we need biomarkers. If not every patient is responding, we need to personalize medicine and predicting response to therapy upfront, which must be included in the clinical trial. We need simple treatment regimens. Hepatitis B is already complicated enough. We have been talking about this. Nobody understands why genotype differs, E positive, E negative. And if I add another level of complexity, it may not help in clinical practice. And I need well-designed trials immediately, including clinical outcome measures. So long-term follow-ups, number needed to treat will be an issue if I get rid of S antigen and have to prove for authorities that I save a life, which will be challenging. And I need long-term data from the very first start of therapy. And maybe my last point, I'm not very optimistic. Will cure from HPV really be possible at all? And we have to keep in mind that I think we are quite naive because we try to overcome 100,000 years of co-evolution. HCV has been, for example, in man, how long? 100 years, around 120 years. We're talking about here for HBV of 100,000 years. This is a paper from Greece when they recovered HBV from this mummy in, I think, South Korea in 1600 something. They compared sequences, and most likely HBV left Africa together with humans 30 to 40,000 years ago. It diverged thereafter. There are so many clever tricks of this virus that has been adapted. The simple fact that it keeps people healthy until the age of 30, 40, until the mothers have done their job, have delivered the virus to the baby, and then when they are getting too old, they are useless, they are just taking resources, and then the virus is killing them. I think it's quite clever overall. The question is really whether we will be able, for example, with immunotherapies to overcome these things. If we target directly the virus, getting rid of this one, this might be possible, but simple immunotherapies to cure HPV, I'm rather skeptical. Thank you very much.